Our first talk is from Abraham Cisneros-Mejorado, who is a researcher in the Cellular Neurobiology Laboratory at UNAM, and will give us a talk about demyelination in a murine model of multiple sclerosis monitored by dMRI and histology.
Okay, thank you. Let me. Can you see the presentation?
Yes.
Excellent. Okay. Please let me the pointer. Okay. Hello, everyone. Thank you for this opportunity to talk with another attempt to verify some findings with MRI and histological data in an animal model. I'm going to tell you about a little about one of line of the research that we have in the cellular neurophysiology lab at the Instituto de Neurobiología in the UNAM in Mexico. This is about the demyelination in a preclinical model of multiple sclerosis, which is monitored by MRI, of course, and also by histology. In this context, we can talk about two scenarios. In a normal condition, we have the transmission of the information in the central nervous system is through the propagation of action potential in the neurons. And in white matter, this is optimal due to the presence of myelin and this lipoprotein structure that surrounds axons and is made by oligodendrocytes. A reminder of what has already been mentioned in today's session. However, in a scenario of damage of myelin, that is a demyelination, occur with almost all of and perhaps all neurodegenerative diseases and some other pathologies, a deficient function of transmission occurs, resulting in impairment motor function or cognitive impairment function also, as occurs in the diseases I mentioned, for example, here. It's very important to mention that these diseases, including the multiple sclerosis, in these diseases, there is no spontaneous or natural repair, at least not in complete manner. So current research focuses and design a therapeutic strategy to promote repair, that is demyelination. In this sense, some efforts are made to promote the oligodendroglial lineage to address this damage, such by recruiting OPCs or oligodendrocyte precursor cells, which could proliferate, migrate, and differentiate into mature oligodendrocytes to remyelinate. In our lab, we have used the enhancement of GABA-A receptors, which participate in myelination. In this way, we have used drugs like the beta-CCB that enhance the activity of these receptors and maybe enhance the remyelination. Next, I will show you some findings in this context in animal model. Represent image of the fractional anisotropy map, employing a diffusion protocol, of course. From cross-section of rat cerebellum are shown in this panel. A region of the cerebellum called inferior peduncle, as indicated in yellow line. We induce a unilateral focal demyelination with stereotaxic injection of ethidium bromide in this area. And we observe that the fractional anisotropy decrease in this region in injured animals, of course. And very important, this matrix is recovered when we employ the strategy for modulating oligodendroglial GABA receptors, like mentioned before. Moreover, these findings correlate well with the histological analysis made with a specific stain myelin. The stain is black gold and stains in red color all myelinated regions. As can be seen in the parasagittal sections, the region of the inferior peduncle is indicated in control, injured animals plus pharmacological treatment. You can see in the lesioned animals a lesser degree of stain. This indicates demyelination, but this is recovered when the animals are treated with beta-CCB. Exists improve of myelination. Now, this showed that it's possible to employ this protocol with diffusion MRI and histology together to monitor both demyelination and remyelination in a focal model. But now we wonder if it will be possible to test this in a model of a disease such as multiple sclerosis. So currently, we have some objectives such to evaluate demyelination by this protocol, the MRI and histology, in a multiple sclerosis model using the cuprizone model. And next, explore the remyelination through this protocol using pharmacological strategies. For this, we have this experimental strategy. We induce a generalized demyelination in mice with the cuprizone model. Cuprizone is a toxin that attacks oligodendrocytes, and then myelin is lost. That is, it induces demyelination. And also, we computing fractional anisotropy, ADC, and diffusivity, axial and radial. And in parallel, mainly at the final point of this experiment, we measured the degree of myelination using black gold staining, which, as told you before, stain in a red color in all regions that contains myelin. The MRI is performed at the National Magnetic Resonance Imaging Laboratory, la NIREM for its acronym in Spanish, and the processing was done in the MRI software, and we used to estimate the tensor model from which we derived the different metrics. Basically, we have mice of C57 strain, 8 weeks old, males, fed with 0.3% cuprizone mixed in the food. With this, we scan the animals before and at the end of cuprizone intake, and after another scan in the 6 weeks after a cuprizone intake. This is 3 weeks of cuprizone continuously intake and 3 weeks more with the restoration of normal diet. Of course, at the end of the experiment, we do histological analysis. Now, I will present you some of these findings. In this panel, you can see the representative image of the principal diffusion vector and the fractional anisotropy maps. These maps is observed at the 0, 3, and 6 weeks after cuprizone intake. At the left, you can see a diagram of the mouse brain in which we analyze the region are indicated. For example, anterior corpus callosum, medial corpus callosum, fimbria, and internal capsule. And also a little bit about the brain cortex in this model. Here is the corpus callosum, anterior and medial. Not some regions, the 4 calculated metrics are shown. Fractional anisotropy, upper diffusion coefficient, and the diffusivities. In both cases, anterior and medial corpus callosum, we found significant differences only in the week 6 post-intoxication with cuprizone. This indicates that cuprizone-induced change in these metrics. These findings are in line with some other that use similar protocols and analyze this same region. Then we quantify the same metrics in other regions because one of the main objectives in this carry-out is carry out a multi-region evaluation in this model. In this way, we evaluate, for example, fimbria and internal capsule. Although the fimbria shows change in the fractional anisotropy from the 3 weeks, the internal capsule shows difference only at the 6 week. Furthermore, we explore this in a region of the cerebral cortex, known better as gray matter, and we were not able to observe significant changes. This suggests that cuprizone 0.3% intoxication promotes change in multiple white matter regions, and that all of these regions are feasible to analyze in the same protocol. Again, this is in agreement on some reports. Then, to verify, then we evaluate with histology the degree of myelination in these regions. In this image, you can see the amplification of regions such as the corpus callosum, fimbria and internal capsule. A lower degree in both conditions, in control conditions and in cuprizone conditions. A lower degree of staining can be observed that there is a demyelination in animals treated with cuprizone. These findings directly correlate with the diffusion MRI metrics in this protocol, the metrics that I showed you previously. However, when we analyze the brain cortex, we observe that, in fact, there are also a demyelination caused by cuprizone in brain cortex, but inclusive in the hippocampus. But in the case of cortex, these do not correlate with the diffusion MRI metrics. This protocol is useful for measuring demyelination in white matter mainly, while it should be improved for gray matter regions. Finally, we also analyzed the inferior peduncle in the cerebellum in this model, in the cuprizone model, and found the following. The image again showed the fractional anisotropy at 0, 3, and 6 weeks after the cuprizone intake. It's possible to observe that the fractional anisotropy decreased in some regions. Here I show you the inferior cerebellar peduncle. As you can see, we found that both the fractional anisotropy, ADC, and radial diffusivity have a significant change from the week 3 of the cuprizone intake, change that remain until week 6. And we then to analyze the degree of myelination with the black gold stain. In this case, I show you amplification of the inferior cerebellar peduncle in a control condition at the end of the experiment. At 3 weeks of the cuprizone intake, some animals were chosen for this condition, and also at 6 weeks after cuprizone intake. You can see how there is a demyelination in 3 and 6 weeks. And again, this directly correlated with the diffusion MRI metrics. In summary, it is feasible to employ a diffusion MRI protocol to monitor demyelination in multiple regions at the same time in the preclinical model of multiple sclerosis, in this case, in cuprizone model. The protocol works well for white matter, while it should be improved for gray matter, at least for brain cortex, probably due to the nature of region where myelinated fibers do not necessarily travel in a parallel manner, while other factors could be involved, of course. And also, the data suggests that there is heterogeneity of demyelination in the central nervous system due to the cuprizone intake. In general, this is very interesting because the findings in multiple sclerosis disease actually present heterogeneity in demyelination, where some of regions are more susceptible to damage and are more susceptible to myelin loss. Now the next challenge, the next step is to analyze the effect of pramyelinated drugs and evaluate whether these metrics in multiple regions are recovered with these therapeutic treatments. Finally, I would like to special thank to the people who directly or indirectly support the continuance of this research, the organizations that support these projects, the sponsors, and of course, thank you for listening to me.
Awesome. Thank you so much. Some very beautifully stained histology. Maybe in the interest of time, if there's one question.
So there's not, but I have one. So I was really wondering, when you look at your results at 0, 3, and 6 weeks, I was surprised to see that at 6 weeks, the metrics are often worse than at 3 weeks, even though you stopped the cuprizone treatment. So I was wondering how you would interpret that. Does that mean that like the damages which happened during the cuprizone treatment started to induce some other damages later on? How would you interpret?
This is a characteristic of the cuprizone model. there is an imbalance between demyelination and remyelination process. When you induce the demyelination with cuprizone, the cuprizone induces demyelination inclusive after the restoration of a normal diet. Why? I don't know. Maybe it's because this imbalance on the recruitment of the oligodendroglial lineage to repair this. But well, it's matter of debate yet.
Thank you. Yeah, I think for a matter of time, we're going to move on. We're a bit behind the schedule. Thank you again.
Thank you.